She is Dr. Jamie O'Rourke. Currently, she works as a research geneticist at the USDA NAMES and is also an affiliate assistant professor at Iowa State University. She earned her PhD in genetics from Iowa State University and her bachelor's degree in biology from the University of Kansas. She served as a postdoctoral research associate at the USDA in Minnesota and as a postdoctoral research assistant at the University of Minnesota. Dr. O'Rourke's research has made significant contribution to understanding how plants respond to abiotic stress, particularly iron and phosphorus deficiency. Her work emphasizes sustainable agriculture, aiming to improve crop yields while minimizing losses from environmental stressors. This morning, Dr. O'Rourke will present the omics of legumes' nutrient stress responses. Thank you, Jamie, for joining us today. Please join me welcoming Dr. Jamie O'Rourke.
Well, thank you so much for that wonderful introduction. And to the organizers, thank you so much for the invitation to speak here. The Baker Symposium is quite an honor. My favorite topic to talk about is soybeans. And so I know that this audience does not need a lecture on the importance of soybeans, but I do want to use this slide to point out just a couple of things. The first is that we really didn't start breeding for soybeans until the 1920s. Before that, soybean was a forage crop. And so in the last 100 years, we've really translated soybean from this sprawling forage crop into the beautiful row crop that we see in the fields today. And in that 100 years, we've readily adopted the soybean. You would be hard pressed to go into the grocery store and buy some prepackaged foods that don't contain some sort of soybean in them. In that 100 years, we've gone from a yield of about 10 bushels per acre to over 40 bushels an acre. But I think that we can do better because I think we're leaving a lot of yield on the table because we lose it to biotic and abiotic stresses. And so today I'm going to talk about iron and phosphate deficiency. And so I am so pleased to talk about the Iron Deficiency Project because it has been a fabulous collaboration between Iowa State and the USDA for over 45 years. You can see that the first paper here was published, I think, in 1979. And iron deficiency is a real problem in the upper Midwest, which, as you saw from the slide before, is where a lot of the soybean growing region is. And it's very environmentally dependent. It has a lot to do with the soil characteristics. And you can see that you get this characteristic yellowing of the soybean. But the problem is that with the interveinal chlorosis of the soybean leaves, for every 1 point increase on the chlorosis scale, you're getting a 20% reduction in yield. Now, there's plenty of iron in the soil, but it's mostly available as iron 3, and soybeans can only utilize iron 2, and so we have a little bit of a problem there. Now, as part of this wonderful long-term collaboration, I want to point out one experiment because it's fundamental to my research, and that was a study by Shunfu Lin and Randy Shoemaker. They took a mapping population, they put it in a field where they know they get iron deficiency chlorosis. They did a QTL study. They took the same mapping population, put it in this hydroponic system, and did a QTL study and found they identified the same QTLs. And that gives me the opportunity to work in my hydroponic system and have a pretty good confidence that what I'm identifying in hydroponics might not be exactly what we're seeing in the fields, but it's a pretty good representation of what we're seeing in the field. So my work is all done in hydroponics. Now, like I said, if you have driven across the upper Midwest in the early spring, you have certainly seen iron deficiency chlorosis in the soybean fields. They are not quite as usually perfect as these plots at one of the fields neighboring one of my family farms. They were running an experiment for iron deficiency, but you'll see these bright yellow spots in the field. Sometimes I almost run off the interstate when I see them because I'm so excited. I'm like, ooh. And like I said, soybean can only utilize iron 2. And so in order to get iron 2, the soybean will release protons from the root system, which acidify the rhizosphere. This will help the iron 3 decouple from other components of the soil. It can be reduced to iron 2 and then transported into the soil. So what about phosphorus? Well, we apply phosphorus as a fertilizer. Phosphorus deficiency is not really a problem in Iowa or in the upper Midwest, but I think it's a pending problem. First of all, plants only take up about 10 to 30% of the phosphorus that we're applying to the soils, so the rest is lost and contributes to the eutrophication of our waterways, so that's a problem. The other problem is that we don't own the phosphate mines. We know where they are, but we don't own them, and experts estimate we only have between 10 and 40 years of easily accessible rock phosphate left. And after that, it's just going to get much more expensive. And since we don't own them, we won't be in charge of that price. So in order to keep farming fiscally feasible, we really need to start thinking about how to develop phosphate deficient tolerant plants. So I worked on phosphate deficiency as a postdoc. I worked in white lupin, the funny little plant on the bottom there. It's a poster child for it. And as I was doing some of my research, I noted that a number of the genes and molecular networks that I was identifying in phosphate deficient lupin were the same ones that I was identifying as a grad student working on iron deficiency chlorosis. And so I thought, I wonder if this macronutrient phosphorus would be inducing the same genes and networks as the micronutrient iron. And my postdoc advisor and I went back and forth on this, but we couldn't answer it. So I just kind of kept this idea in the back of my head. And when I started here with Iowa State and the USDA, I decided to test it. And the first thing I needed to do was see if soybean even responds to phosphate deficiency, because again, we don't study it because we just apply it as a fertilizer. And so you can see in this image that I systematically remove phosphate from my hydroponic system. And even at 75% phosphorus, you can see there's a dramatic change in the root architecture. By 0% phosphorus, the roots look completely different. They're thicker, there's more roots up at the top. And unlike Arabidopsis, rice, and white lupin, soybean roots get longer. And that's going to be a theme. Soybean doesn't always play by the same rules that model species play by. So if there is a phenotypic change, that means it must be induced by transcriptional changes, and that is my wheelhouse. And so I designed what I think is a fairly elegant experiment. I'm still pretty proud of this one because conducting it almost killed us. So we started plants in rolls, we transferred them to our hydroponic system, we put everything in control conditions, everything was looking great for 8 days. After 8 days, we transferred plants. Some went into new control conditions, some went into iron, that's the yellow, some went into phosphate deficient conditions, that's the pink, and we left things for 24 hours. We took a subset of plants. We moved everything back into control conditions for 48 hours. We took a subset of plants. We moved our plants again to either a repeated iron stress, a repeated phosphate stress, or a late stress to account for the difference in age between the two stress time points, or a sequential stress exposure. After 24 hours, we took the remaining plants and we extracted RNA, sent that off for RNA-seq. While we were waiting for that, we did this again, and this time we left our plants and we collected some phenotypic data. Plant breeders are great at reading line bar graphs, so I am not going to go through those. But needless to say, the plants that were exposed to any period of abiotic stress, early stress, repeated stress, late stress, seem to be healthier, stronger, more resilient plants than those that were grown in control conditions the whole time. And that's kind of counterintuitive to what we would think. We always think of stress in the fields as bad, but it turns out, I think, short periods of abiotic stress are actually going to be beneficial to those small seedlings. So I got my RNA-seq data back. I went through my pipeline, I identified all the differentially expressed genes, and now I can answer my question, are iron and phosphorus deficiency utilizing the same genes and pathways? And the answer is kind of. At the first two time points, the genes are really unique to the two different stresses. They do not share a lot of the same genes, but at the later two time points, which is actually one time point, just either repeated stress or late stress, the majority of the genes are differentially expressed by both iron and phosphate deficiency. And so I called my postdoc advisor and said, well, I guess we were both right. If you look at the number of differentially expressed genes, that's too many to look at one by one, which is what we would do in Arabidopsis. But in my lab and our group, we choose to look at all the genes that are differentially expressed. In order to do that, we use heat maps. Now, I think the Baker Symposium has seen a lot of heat maps, but just to walk you through it, the ones on the far side are iron, the ones closer to me are phosphate deficiency stress. Each column within the heat map represents one of my time points. Leaves are on top, roots are on the bottom. If they are blue, the genes are upregulated, yellow, they're downregulated. I made these 4 heat maps, and the first thing we can see is that there really seems to be a shift in expression between the first and second time points and the last 2 time points. We went back, we checked the data, we've rerun the analyses. That's just a biological reality. We think we've spanned some sort of important developmental process in the seedlings with our experiment, unintentional, but this is exactly what the expression levels look like. The other thing we noted, and you can see it just by looking at the number of differentially expressed genes, if you have really good eyes, and gotta say, I can't read that from here, is that the number of genes are very different. Soybean seems to be responding to that iron deficiency stress at that first time point, but it doesn't respond to that initial phosphate deficiency stress. It does, however, induce a massive transcriptional response upon that iron or phosphate recovery. So once we put the plants back into those control conditions. And I thought about this, and I thought about all the work I did at phosphate as a postdoc, and I thought, you know, this probably actually makes sense, so let's just test it. And it does make sense. Phosphate is a macronutrient. There's a ton of phosphorus stored within the plants, and one of the biggest places it's stored is in the phospholipid bilayers. All the membranes in the plant have that phosphate group in the head, and the plant can scavenge that phosphate group, replace it with a galactose group. So now you have galactolipids. They're less flexible, they're less permeable, but they serve their purpose, and they provide a pool of phosphorus so that the plant can maintain cellular activities. We know the biosynthetic pathway to make this happen. If we look at the gene expression of the genes associated with that pathway, we can see they're upregulated in leaves and roots. I think the two that are not in both is really just a matter of the timing of my samples. So I could go through and I could look at a lot of these biosynthetic pathways, but again, with 13,000 genes, that would take me forever, and that would just not be very feasible, especially when you have to publish. So again, we went back to our heat maps. This time we used some statistics to identify clusters of genes that share the same gene expression profile. And by that, I'm not saying like they're expressed the same in all 4 time points. I'm saying like all the genes in one cluster share the same expression profile across all 4 time points. So maybe they're all upregulated at the first 2 time points, all downregulated in the second 2 time points. That's most easily seen in the roots. So we have these clusters, and now we can start to look at the annotations of the genes associated with each of these clusters. But again, some of these are really big. And so we chose to look at the gene ontology annotations associated with these clusters, and not just which gene ontologies we had, but which ones were overrepresented. And so again, we used some statistics, identified overrepresented gene ontology terms. We graphed them. That wasn't very informative, so we transformed our graphs and did some data transformation. And we came up with this. I call this my mountain graph. And basically what you can see with this is that the genes and networks utilized by iron and phosphorus really appear to be the same. The 2 panels, iron on the far side, phosphorus closer to me, the trends appear to be basically the same. Once again, you can see I've put the dashed line at the recovery time point. So in a number of cases, you can see there's a big dip in iron deficiency gene expression at the initial stress response, but the big dip comes in the recovery response for phosphorus. But they're basically utilizing the same trends, which really means that they're utilizing the same molecular pathways. The other thing that we noted here is that if we look at the annotations associated with the clusters, and again, you can see that the gene expression for the genes in these clusters is pretty conserved, is that the annotations are fairly conserved. And I've highlighted the annotations in bold that are annotations we would expect for the Clark iron deficiency response. And Clark is the line we've used for over a decade to do our iron deficiency experiments. And it really has a trifecta response. We would expect defense responses, nutrient stress responses, DNA replication, and methylation. You can see those are really highlighted in bold here. Looks like phosphorus is utilizing the same responses. The final thing is that roots seem to respond faster. That makes sense. They're in the hydroponics, they're exposed to the stress deficiency, and then that signal has to get up to the leaves, has to be interpreted, and the response has to be induced. And so the roots seem to respond faster and in a more concise manner. The other thing we can learn from this, and something that I've always been interested in, are transcription factors. And so I don't know if you can read it, you can't. The transcription factor families are the x-axis on the bottom, and then the counts for the transcription factors in each of the samples is on the y-axis. It's again divided by the 4 tissues, so leaves are on the top, roots are on the bottom, iron and phosphorus. And then the counts of the number of transcription factors in each of those samples is the line graph. And what we've done here is we've counted them by the GO categories. And so the first time I looked at this, I thought, oh, this is going to be great. We can identify peaks that are unique to each of these panels. And there just really aren't that many peaks that are unique to each of these panels. And so we looked at this again and thought, you know what we can do, and that's really more interesting and better for us, is we can now dissect the big transcription factor families. And so I've highlighted AP2, MIBs, and Werkies. And so these are massive families. We always see members of these families differentially expressed in our experiments. And for the first time, I can say, well, these 50 AP2, these, I can't read it, the 38 MIBs and 24 Werkies, and I've got those numbers wrong, but are associated, the top line there that's bold, are involved in defense and immunity. If I look at the green line, I can say that these 18 AP2s, these 4 MIBs and 7 Werkie transcription factors are involved in photosynthesis. And that's not something we've ever been able to say before. And in fact, this has become a really excellent resource for our lab group because you'll see in some later experiments, we don't always get the vast number of differentially expressed genes that we did in this experiment. But now we can go back to this as a resource and say, but what, at the same time point, so what was that transcription factor doing in our earlier experiments? And it's probably doing something very similar in our later experiments. So this has been a really nice resource. So what about that sequential stress response? Plants that were exposed to iron deficiency and put back in control conditions and then exposed to phosphate deficiency. Well, it took us a long time to figure out how to think about that. And we decided the best way to think about it was to compare it to gene expression to the two repeated stresses, either repeated iron or repeated phosphorus. Again, plant breeders are great at looking at diagrams, so you can see the Venn diagrams. I prefer to look at heat maps. These are made in a slightly different manner. But what you can see is that the two tissues respond very differently. The majority of the genes that are differentially expressed in sequential stress in leaves are unique to that sequential stress. The majority of the genes differentially expressed in the roots are differentially expressed in repeated stress and sequential stress. And interestingly, their expression profile is conserved. It's not just that they're differentially expressed, they're consistently differentially expressed in those three stresses. So we went through and we identified all the possible expression combinations we could expect to find. That's the middle panel, and we counted the number of genes. But really what I want to point out is that the core genes that are conserved, and it's very obvious in the roots, but there is a suite of core genes conserved in the leaves. In leaves, those are involved in cytokinin signaling. In the roots, it's that trifecta response that I just described earlier. We've also identified genes that are important for phosphate deficiency, right? If you've exposed the plant to just iron deficiency, those genes are not differentially expressed, but any type of phosphate deficiency, you get those. Those genes are involved in galactolipid biosynthesis, like we talked about, and LRRs. We identify first stress signature genes, genes that are expressed under iron deficiency stress, not under phosphate deficiency. And those are really related to phytohormones. And so the final thing we identified with this is we know that plants respond faster the more they're exposed to different stresses. And we think this shows that the plants are not just increasing the expression of already differentially expressed genes, but that each time they're recruiting new genes to that response, and that's what's helping them respond faster. So I could actually go on and on with heat maps and Venn diagrams, and if you want more, please stop by my office. But this is a plant breeding symposium. So how do we translate what I'm doing in hydroponics and in greenhouses and growth chambers to the field? Like, how are we going to make this applicable to plant breeding? And the best way we figured out is to really leverage classic QTL studies. And so there's a QTL on chromosome 3. It's responsible for about 74% of the phenotypic variation. It was identified a number of years ago. We know that in Arabidopsis, there's a transcription factor, basic helix loop helix 38, that is the master regulator for iron deficiency chlorosis. It happens to be located, the soybean homolog happens to be located in this QTL region. But if we look at the expression profiles of the genes in that region, so here we have all 490 genes in this QTL region, and then the expression profiles are in the little bubbles off to the side. The dashed line in the zoomed in component of the slide represents that basic helix loop helix transcription factor. And you can see it's not really differentially expressed. Now, maybe it's the timing of these samples, but we've tried silencing it and we really don't impact the iron deficiency stress response of soybeans. But there are some genes really close by that are very differentially expressed. Interestingly, we don't know what they are. Soybean has a lot of genes with unknown domains in them, but they clearly are playing a role in the iron deficiency stress response. Further down in the QTL, we can see some ethylene response transcription factors, very differentially expressed. So these might be good candidate genes to underlie this QTL. We can do the same thing with phosphorus. Luckily, a couple of groups in China have identified the phosphorus efficiency QTLs. They don't really overlap with soybean, or with iron, not surprising. They're much smaller. And so if we look at the genes here, there are some genes we would pick out just based on their annotations: phospholipases, acid phosphatases, SPX. If you work in phosphorus, these are genes you would think that's the gene. That's got to be it. But if you look at the expression profiles, there are a lot of other good candidates in there, and some of them really make sense. We've got LRRs, we've got some unknown genes, but we've got some heat shock proteins and some RLPs. These are genes that we might not consider, but if you start to look at genes underlying QTLs and that are responsible for some of these disease traits, these start to make a lot of sense, but we wouldn't have picked them out for phosphorus. And finally, we can kind of put all of this together. And this is a really beautiful study, and I really have Danny Singh's group to thank for this. Again, going back to that collaboration with Iowa State and the USDA. Danny Singh's group took 400 plus PI lines. They put them in the field where they knew they would get iron deficient conditions. They put them in hydroponics where they knew they could induce iron deficient conditions. And they ran a GWAS study. As you can imagine, they identified dozens of regions in the soybean genome that are important for IDC, including the region on chromosome 3. But when we looked at that region, we said, that looks a little odd. And so we asked Xiaoping to go back and look at this region. He was doing the bioinformatics. And he came back to us and he said, actually, that's 4 distinct linkage blocks that are all stacked on top of each other to form that QTL. And when we looked at the genes in that region, we could identify genes related to our soybean trifecta response in each of those 4 regions. And so right away, we know that this is an excellent finding. Like, this is really going to transform how we think about this QTL. But we followed up on that in a number of ways. One, we took some of the lines, we selected them for their iron efficiency based on their SNP profiles. We put them in hydroponics and we screened them for how fast they respond to iron deficiency stress. And we identified lines that respond faster than Clark, our standard line. Clark already responds so much faster than Arabidopsis. So these lines are really fast responding lines. The other thing we did is looking at the annotations of these genes. We also looked at the expression pattern of these genes. And Michelle Graham had a graduate student, Daniel Colhays, many of you know him. And he, based on some work by Chantal McCabe, said, well, let's silence these genes. And so he silenced them individually, didn't affect iron deficiency. When he silenced them together, and it's GLU1 and ARR4, and you can see they're highlighted, when he silenced them simultaneously, he did impact iron deficiency, which leads us to believe that this QTL is a multigenic trait. There's multiple genes within this QTL. We don't think it's just those 2 genes. We think it's lots of different combinations of those genes. So this is great. We really need to understand how this QTL works and how iron stress tolerance is affected. But the problem is this QTL is really fixed in the elite lines. And so we really need some novel sources of resistance. And like I said, between Danny's group and a lot of other groups, we've identified a number of regions in the soybean genome, but which one should we go after to identify some new regions for resistance and resilience? Well, about 13 years ago, a couple of plant breeders screened a number of these PI lines, and they identified FISCB3 is resistant to basically every stress they threw at it. This little plant it's the little engine that could. It just keeps chugging along. And it was identified from FISCB Sweden. So it's a northern line. It's actually a 000 line. So my collaborators in Minnesota, Bob Stupar and Aaron Lorenz, thought this might be a good line to start integrating into their breeding program. So they made some crosses. They sent them to me. I put them in hydroponics. They segregated for iron deficiency. So they put them out in the field. And they were able to do a QTL study. And they identified a region on the top of chromosome 5 that is responsible for the FISCB iron deficiency stress response. Now you can see we've identified this region in other studies, but it was always responsible for such a small portion of the phenotypic response that we didn't pay it any attention. But in FISCB, it's responsible for the majority of the phenotypic response. This is great. Bob and his group did some fine mapping. They got it down to 17 genes. We were on a conference call and trying to decide, like, well, which one of these genes, based on the annotation, do we think we should knock out for CRISPR? And I said, well, why don't we try and silence all of the genes individually using VIGs, and that should give us a pretty good indication. Guys, they bought it. So we did that. And you guys know how VIGs works, but I just want to remind you, you put a segment of your gene into this viral vector, and now the plant is going to try and destroy anything that looks like the virus, including the sequence of your native gene expression. And so, again, there's 17 genes. We determined 15 are transcriptionally active. On our first pass, we were able to develop 10 constructs that worked right off the bat. When we sequenced them, they were what we thought they were what we thought they should be. And so we decided to go ahead and start our experiment. So on the bottom, you can see we have plants infected with just the buffer, plants infected with just the vector, and then plants where we've silenced a single gene in each one of those pots. Obviously, if we silence a gene, we're going to get stunted plants, but those plants all look really healthy, except that plant in E. And that plant in E has very characteristic chlorosis responses. I immediately called Bob and Aaron, and I'm like, guys, I got it. It's this gene. So we wrote a grant, and USB was fortunate. We were very fortunate that USB chose to fund us. And so we continued with our experiments. We took Fiscabee, we put it in hydroponics, we infect it with our vector. And again, under iron sufficient conditions, we got exactly what we were expecting, got chlorotic symptoms. But under iron deficient conditions, the plant looked just like the controls. That was unexpected. But we have this grant and we are going to continue our experiments and see what we can find. Now, as we were doing this, we started looking at what this gene is. Turns out it's a DTX mate transporter. It was one of our candidate genes. We really thought it might be based on the fact it was a mate transporter because mates allow plants to respond to biotic and abiotic stress. And this is DTX. In cotton, if you overexpress it, it actually confers abiotic stress tolerance. So again, we think this is great. So we go ahead and we run our big hydroponic experiment, which in comparison to the last one, you can see it's not as big. We chose to include Fiscabee that was not infected with anything because we don't know anything about the Fiscabee stress response, right? This is totally new and novel. We have plants infected with the empty vector. We have plants where we silenced our mate gene, grown in iron sufficient and deficient conditions. We grew the plants in hydroponics for 14 days because we have a wealth of studies in Clark at 14 days and we get thousands of genes differentially expressed. And we thought we want, on this first pass of experiments, the biggest picture of this Fiscabee stress response possible. Well, you can see the number of differentially expressed genes we got. It was not many, guys. And so right away, when we started doing the gene expression data analysis, we knew that this was just going to be very different than what we had seen before. So starting with the uninfected Fiscabee, I'm not going to go into the details, but needless to say, this responded exactly the way we would expect a plant to respond based on the studies in Arabidopsis. Now, because we did not have very many genes differentially expressed, I did look at all of them. And there was one gene that stood out to me, fraxitin. Fraxitin is a protein, it's exuded from the roots, and it actually will extend the pH range so that the ferric reductase can more efficiently reduce iron 3 to iron 2. We'd never seen it before in our experiments, and so I thought that was actually pretty interesting. If we start to look at the genes differentially expressed between the empty vector and the VIG silence plants, we start to see a different picture. So this is the 228 genes differentially expressed in the leaves under iron sufficient conditions. I can make the same kind of analyses of the 69 in iron deficient conditions. It looks pretty similar. I've taken the Arabidopsis homologs and I put them in the string DB, which looks at protein interaction. So how do these genes interact with each other? There aren't very many. And you can see that a lot of them do interact. You can see that we have genes involved in RNA processing, the general stress response, photosynthesis, and then iron and phosphate responses. Based on my previous work, you can say, like, that makes perfect sense. Except if you look at these genes, they're really all phosphorus genes. The only two that are really iron specific are the two that I've boxed. It's Brutus and NRAMP3, and they're expressed in the opposite direction that I would expect. And so we kind of started to hypothesize that maybe by silencing this gene, we've upregulated phosphate deficiency stress responses, or the plant is misidentifying the stress. Could go either way. The other thing I want to point out that isn't part of an image, but one of the 18 genes in the leaves that's differentially expressed between iron sufficient and deficient conditions is actually the basic helix loop helix transcription factor. But that gene is not expressed at all in any of the VIGS plants, not just differentially expressed, it's not expressed. So by silencing the mate gene, we have silenced the expression of that basic helix loop helix transcription factor. What we think we might have done is induce some backup systems. So IRT3 usually transports zinc, but under stress, it can transport iron. And so again, these are just really novel responses. And so USB said, well, let's learn some more. And so we did another experiment. This time we grew plants. We thought we maybe shouldn't grow them for 14 days in the stress conditions. That didn't give us very much information. And so our plants were all harvested at the same time, but you can see they were exposed to either 1 day of stress or 7 days of stress. We again have our silence plants and our empty vector plants. And this allowed us to make, so again, some really interesting comparisons. And so I could walk you through the heat maps, I could walk you through the Venn diagrams, they all look pretty much the same. So the highlights for you is that in the leaves, over 50% of the genes related to iron deficiency between iron stress and control conditions are related to phosphate deficiency. If we look at 1 day versus 7 days of iron stress, the longer the plant's exposed to the stress, the more phosphate stress genes you see differentially expressed. And then finally, if we look at the treatment, the empty vector to our silence plants, there was one gene differentially expressed in all but one comparison, and it just barely missed the statistical cutoff for being differentially expressed in that one. And it was MED5A, 5B. Now, I normally would have just ignored that, except again, I've always been interested in transcription factors, and I happen to know that MED5A, 5B is part of the mediator complex. And in fact, it is the part of the mediator complex that directly interacts with transcription factors that are bound to the promoter region. And so I thought this was really interesting. I started to look at this, and in Arabidopsis, you can see if you knock that out, you increase phenylpropanoids. It also may regulate defensin 1, which has some other implications for nutrient deficiencies. But the really interesting thing about this is that soybean has a duplicated genome. MED5A, 5B, the differentially expressed gene, is actually glymin19G 1500. Yeah. But for every gene in the genome, there's a duplicate. And the duplicate of 19G 1500 happens to be right next to our gene of interest, our 5G 1700, our mate gene. And so it turns out it was actually would have been silenced by our construct D. And so perhaps I didn't overwater that pot. Perhaps it was actually a phenotypic response. And so maybe the chromosome 5 QTL, like the chromosome 3 QTL, is regulated by multiple genes. We don't know for sure yet. We're still working on it, but we have designed a vector to silence both 1600 and 1700 simultaneously. It would silence that gene on chromosome 19. If we put it in Fiscabee, silencing those genes in soil, we really don't see any phenotypic differences. But when we put it in hydroponics, we do start to see differences. But again, maybe not the differences we were expecting, but we've only done one round of this, so we clearly need to repeat it. The other thing I want to point out that we didn't think to measure, because we weren't expecting it, was that the leaf area size is dramatically different when we silence the two genes together versus a single gene. And again, we need to go back and look at some of the data from Daniel and some of the data from Chantal's group to see if this happens just because we've silenced two genes, or if it's a combination of silencing these two specific genes. So clearly we have some more work to do on this, so I will try and get back to you with an answer. We've got some experiments running right now, but I think we're probably on the right track. And so just to sum up, and the take-home messages, again, this is preaching to the choir, but if we want to understand stress responses in crops, we need to study stress responses in crops. And we can't just study one line at a time. Ironic, I know, because I just talked about Fiscabee where I was just looking at one line at a time, but really, we've been held back historically by computational resources where it was impossible, but now we have the resources, we can look at multiple lines, we can look at multiple time points. The soybean germplasm contains multitudes, right? Danny's experiment showed us that there's not just one way to achieve iron stress tolerance. There's going to be lots of ways within the soybean germplasm to achieve any sort of trait you want, but it's going to take collaborations between plant breeders, geneticists, and plant physiologists because these responses are complicated and we all have our various areas of expertise. And so it's only going to be through those collaborations, like the collaboration with Iowa State and USDA, that we're going to identify those. And finally, I just want to say that I think it's likely that the chromosome 3 QTL for soybean, for iron deficiency, the QTL underlying brown stem rot, that's multiple genes that Chantal identified, and likely the chromosome 5 QTL for iron deficiency in Fiscabee are not outliers. I think it's likely due to the duplicated nature of the soybean genome that in soybean, maybe specifically, that the QTL are really going to be more polygenic traits. And I think that's something we need to consider as we're starting to try and design our future crops. And so with that, I just want to thank the people that have made this possible. My technician, Mike, who did all of the bench work for the Fiscabee research in my group. I could not do any of this without my co-scientist in the USDA, Michelle Graham, and the members of her lab, Lori, Jody, and Daniel, also Chantal. I have fabulous collaborators at the University of Minnesota with Bob and Aaron, the members of their groups, MJ and Ryan, who have worked on the Iron Project with us. And then, of course, the best collaborators are always here at Iowa State. So I really want to thank Steve Woodham's group with the VIGs. We still have lab meetings together. And Danny's group, and the members of his group that we've worked closely with are Zhao Ping and Tashale. And then I'm really fortunate that our group's been funded by a number of sources. The USB, of course, funded the FISCA-B work, and the majority of my funding usually comes from the USDA. So with that, thank you, and I'm happy to answer any questions. I'm also really happy to see some other people have made it in. That's really great.
Okay, hello, yeah. Okay, thank you again, Jamie. If we have any questions, we have 4 minutes.
There's one right behind you.
It's really interesting.
Thanks.
These sites of multifactorial stress studies are really intriguing. In your RNA-seq experiment description, you had an iron stress followed by a phosphorus stress. Did you do the reverse order?
Oh my gosh. It didn't occur to us at the time that we should do that because we were interested in the one. And I so wish we had, but we didn't. And we just haven't gone back to do it again. But I really wish we had. So, let that be a lesson to you. Think ahead. But we didn't.
Yeah, I'm curious. Because you find such an overlap between the two stresses, pardon me, do you think that perhaps experiencing one stress is priming the plant to respond to another stress? And have you speculated on what you might see if you had done the reverse experiment?
That's exactly what I think is happening. Yeah.
Okay, my first question is, how can we link the phosphate deficiency and the phosphate use efficiency? Deficiency and efficiency.
Yeah, so that's a really big area of study is how can we increase the phosphorus efficiency of the plants, and how can we maybe use this kind of data to increase phosphorus efficiency? It's a huge area of study, and I think one of the places we need to look at is how does phosphate deficiency alter the root architecture? Because you can see that it clearly does. What are the genes involved in modifying that root architecture? Because we know if we can get those cluster roots that we see in white lupin, that we could be more efficient at scavenging phosphorus. And then there's a whole nother field that we need to look at, and that's how can we make plants better able to reutilize the phosphorus that's within the plant, right? Better at the galactolipid biosynthesis, better at scavenging it from the vacuoles where they store it. And so I think there are a couple of components that we can look at, and I think we can use some transcriptomics to start to look at both of those.
Okay, thank you.
Thanks.
One question from online, so I'm just.
Sure.
Hi, Jamie. Thank you for the wonderful presentation. Your data has shown that there are a lot of crosstalk between P and iron deficiency, and that there are upstream level TFs seemingly common to both deficiencies. Scales for both deficiencies don't overlap much, likely limited to by population ? Program, it is more efficient to target upstream target like TFs and utilize gene editing techniques, or to target QTLs, which might be genes more downstream and population.
Man, that is a really good question. It is a debate that Bob and Aaron and I have had repeatedly, which genes they should go after. And we don't know what the answer is. The problem with trying to go with the transcription factors that are upstream is you have to figure out which one it should be. And you can see, like, we often get lots of transcription factors differentially expressed. We've done a lot of promoter analysis, which I didn't put in this talk, to look at which transcription factor binding sites are in the promoters of the differentially expressed genes to see like what might be inducing them to be differentially expressed. But I think we need maybe different types of experiments. And we've proposed to do some of those, but I don't have the results from them, where you're figuring out which transcription factors are binding to know which ones to go after. I don't think based on this data that we can know that, but it's certainly an ongoing debate. And I think we'll probably figure it out, but we don't know yet. Thanks.
Okay, so we are on time, so let's thanks Dr. O'Rourke once again.
Thank you so much.